Hey everyone, what's up? Welcome back. So for this video, we're gonna start going into chapter 2, which is basically, it's typically called the transmission genetics chapter. So transmission genetics. And so basically what this chapter is looking at is how are these genes, how are these heredity materials, how are they passed on from one individual to another individual? And typically what you're going to see in this chapter, or from like other courses as well that I teach from other universities, is where you're going to look at is how multiple genes pass on from one generation to another, and some statistical tests on how to confirm your hypothesis in terms of how in terms of like the mathematics behind how you can calculate the different proportions between genes and stuff like that. So this story kind of begins all the way back in the day with Gregor Mendel. So this guy, he was like a monk, and like all monks do, they end up playing around with plants. More specifically, he played around with pea plants. And what he wanted to do with these pea plants is he basically wanted to figure out that whole entire question is, how are these traits passed on from one generation to another? So that was his whole entire goal with the project. And he published all of his findings, but he didn't really get any recognition for it until like after his death. And because of the recognition and because of his work, he's typically referred to as the father of genetics, which is a pretty cool title, not going to lie. So like where do we all begin from this story? So back in the day, they used to believe a lot of theories as to how you pass genes or traits from one individual to another. The most popular theory was the blending hypothesis. So the blending theory of heredity. So all this theory means is where the offspring of two parents will be phenotypically a blend between parent 1 and parent 2. Phenotype. So basically, if you think about this, we would have, let's say we have a blue flower. So let's say we have a blue flower and we cross it with a flower that is white. So back in the day, what they used to believe is that if you made this cross, what will happen is that your offspring are all going to be some sort of a blend. I don't know what color this is going to be honest, and I don't have that many colors on option. So let's say the blend between blue and white is green, okay? So it's going to be a blend between the phenotypes. So it'll be something like that. So this is basically all the hypothesis is stating, is that your offspring is going to be a blend between parent number 1 and parent number 2. So this was like the most popular hypothesis for genetics at the time. And the reason why it was pretty popular is that because you know, like if you look at human heights, you know, if you have a parent that is pretty tall and a parent that is really short, you know, you should obviously expect to see that their child is going to be somewhere in the middle. So that's one of the reasons why this hypothesis was pretty popular. But another important thing to look at when you're looking at this hypothesis is that what would happen if we cross two green flowers together? So what would happen if we cross these together? So let's say we cross these together. What would you expect? So what would you expect is something you would get another green flower. So because of this, if you can determine this, the original blue and white phenotypes, are lost in the next generation. So this is something to keep in mind then with blending theory of heredity, where initially you get a blend, and if you cross that blend together, you would expect to see the exact same offspring again, and you shouldn't expect to see that you would get a white flower and a white flower at all. Or, excuse me, a blue flower and a white flower. So what Gregor Mendel did, he sort of did the exact same experiment, where he took something called a true breeding, blue flower, not Rootley, he used a different flower, I forget to be honest. He took a true breeding blue flower, and he crossed it with a true breeding white flower. So all a true breeding flower or a true breeding individual running out of space. So a true breeding individual is essentially where you would cross the same individual together over and over again, such that your final phenotypes will always be the exact same what you started with. So you're basically crossing blue flowers with blue flowers over and over and over again until they're considered true breeding. Same thing for white. And basically the goal of true breeding is, as I referred to before, something called a genotype. The goal of true breeding is to have homozygous individuals for a homozygous genotype. So that was the whole goal of essentially what true breeding is. And you probably don't even need to remember like the entire process of how he gets to that. You just need to know that true breeding is essentially a person that is, or individual that is homozygous for a certain allele. So what he did was he took these blue and white true breeding individuals and he crossed them together. He ended up with an unexpected outcome where he got blue flowers. All of them were blue flowers. So he had a 100% chance to get blue flowers. So that was kind of weird, right? And what he referred to this generation as the F1 generation, the filial generation. And what he referred to the parents, he referred to the parents as the P generation. So what he did was he took that blue flower from the F1 generation and he crossed it together again. So he took the F1 generation flower, which is the same blue one, and he crossed them together. Let's imagine this one is another F1 here. So he crossed these together. What he ended up with is a 3-to-1 ratio, or basically 3/4 of your flowers are blue, and then 1/4 of them are white. So because of this, he sort of broke the assumption of the blending theory of heredity, where you actually regenerated the parents in the F. This is called the F2 generation, where you regenerated the original parental phenotypes in the F1 or in the F2, and then in the F1 right here, we did not have a blend between blue and white like how we thought there would be a blend. So this was basically his whole entire experiment. We're going to go a little bit more in detail into that later. Another thing that the textbook likes to go over is why did he use p-plans? So he used p-plans because of something called a dichotomous trait. So basically this is just, you would only have two possible phenotypes, two phenotypes only. And he was mainly interested in this because it's pretty easy to study two phenotypes rather than like 500 phenotypes that you could possibly find in nature. Another thing is, with plants, you can also control their fertilization, and you can also self-fertilize them if needed. So you can control fertilization to generate those parental genotypes, or to generate the true breeding individuals. So what we're going to do is, in the next episode, we're going to cover how he specifically did this in chapter 2. And like what, we're looking at one trait here, but how did he get to this, and what conclusions has he made from this? But that's basically what we're going to be doing. Just remember the original most popular hypothesis at today was the blending theory of heredity, and just also understand what true breeding and how did he get these, and how does this data that he generated differ from the data that we've expected from the blending theory. So that's pretty much it. Thank you for watching.